All right, welcome. We're jumping straight into the clinic today with a hands-on challenge. We're going to tackle one of the most common and, frankly, most critical lab abnormalities you'll ever see, the high anion gap. So, let's get our thinking caps on and dive right in. Okay, so picture this. The labs come back, and that number is staring you in the face. The anion gap is high. This is a huge signal that something is seriously wrong. It's screaming metabolic acidosis, but it doesn't tell you why. And that's your job, right? To play medical detective.
So first, what exactly is the anion gap? Well, it's not something the lab measures directly. It's a calculation. You can think of it as representing all the unmeasured negatively charged particles, or anions, floating around in the blood. Normally, that gap is pretty small, somewhere between 8 and 12. But when that number starts to climb, that's a massive red flag. It means some new, unmeasured acid has shown up and is consuming bicarbonate.
When you see that high gap, your brain should immediately jump to these four main culprits. I call them the big four. Are we talking about ketones? Or maybe a problem with lactic acid? Could the kidneys be failing? Or did the patient ingest some kind of toxin? This framework is basically our roadmap for this quiz. You ready for the first case?
All right, challenge number 1. This is a real classic toxicology puzzle. It all comes down to putting two key lab values together to crack the case. Let's see the details.
So, here's our patient. The context, being found near automotive supplies, is a huge clue, obviously. But the lab data is where the diagnosis really gets locked in. We've got our expected high anion gap, sure, but the real clincher here is the high osmolar gap. What that means is there's some substance in the blood that's osmotically active that we aren't accounting for in our standard calculations. Okay, so take a second here. You've got a high anion gap, a high osmolar gap, and a patient who is near things like antifreeze or windshield washer fluid. What two toxins should immediately be at the top of your list? And there you have it, methanol and ethylene glycol. Methanol is what you find in things like windshield washer fluid, and ethylene glycol is the main ingredient in antifreeze. They are both infamous for causing this exact clinical picture. But the real question is, why do they cause both of these gaps?
The key is all about timing. See, initially, right after the person drinks it, it's the parent alcohol, the methanol or ethylene glycol itself, that's circulating. These are tiny molecules that are osmotically active, so they raise the osmolar gap first. It's only later, as the body metabolizes them into their nasty toxic acid byproducts, that the anion gap starts to climb. It's a race against the clock.
Awesome. Let's move on to our second case. This one involves one of the most common medications on the planet, and it presents with a really distinct, almost paradoxical acid-base signature.
Okay, all the clues are right here. We've got a massive overdose. We have our high anion gap metabolic acidosis, but look at the other findings. Ketones are negative, so that kind of pushes diabetic ketoacidosis down the list. But the most important clue, the blood gas shows a simultaneous respiratory alkalosis. So the patient is breathing super fast, blowing off CO2, at the exact same time they have a metabolic acidosis. What on earth does that?
You know, this mixed picture of a high anion gap metabolic acidosis plus a primary respiratory alkalosis is like the classic fingerprint of one specific toxicity. Any ideas?
Yep, you got it, aspirin. Salicylic overdose is notorious for causing this dual disturbance. It's a true acid-base emergency. And the mechanism is just fascinating. Salicylates basically launch a two-pronged attack. First, they directly poke the respiratory center in the brainstem, which makes the patient hyperventilate. That's your respiratory alkalosis. At the same time, they completely mess up how our cells make energy, which leads to a buildup of other acids, like lactate. And what's so scary is that the fast breathing can sometimes hide just how severe the underlying metabolic acidosis is.
Okay, for our final case, we're heading into the ICU for what is, without a doubt, the most common cause of a high anion gap acidosis you'll see in critically ill patients.
The picture here is pretty grim, but it's one we see all too often. A patient with a severe infection, sepsis, is now in shock. Their blood pressure is in the tank. Their arms and legs are cool, all signs of poor perfusion. And, right on cue, the labs show a brand new, severe, high anion gap acidosis.
So this is a two-part question. It's not enough to just name the acid that's building up. What's the specific classification of this acidosis, and why exactly is it happening when a patient goes into shock? And the diagnosis is type A lactic acidosis. That type A part is absolutely crucial because it tells us the root cause is circulatory failure.
So type A is all about a failure of oxygen delivery. In septic shock, the body's circulation just can't get enough oxygen to the tissues. To survive, the cells are forced to switch from their normal, efficient aerobic metabolism to a very inefficient anaerobic process. And the waste product of that process? A flood of lactic acid that just overwhelms the body's ability to cope. So let's just do a quick recap of our 3 challenges. In case number 1, the big clue was that high osmolar gap, and it pointed us straight to toxic alcohols. For case 2, it was that really bizarre mixed picture. The respiratory alkalosis plus the metabolic acidosis, that was the unique signature of aspirin toxicity. And in our last case, the clinical picture of shock and hyperperfusion led us right to type A lactic acidosis.
You have totally nailed the basics of pattern recognition for HAGMA, but here's the thing. It can get even more complicated. Patients can have 2 metabolic problems at the same time. To uncover those hidden, mixed-up disorders, you have to go another layer deep. You have to compare the change in the anion gap to the change in bicarbonate. But that's a whole other level of detective work and a topic for our next explainer.